So welcome to MedSearch Course Mastery YouTube channel, a place where we internalize, medical concepts and concerns in piecemeal. Today, we're looking at histology in anatomy, and the topic we're looking at is inclusion. Watch this. We said as simple as A, B, C. If you can count 1, 2, 3, you should be able to understand the concept. So, if you can simplify it, if you can understand it in a simple way, if it can be done little by little, if that's the way it will stick or be retained, then let's do it. So, for inclusion, we are saying just 3 things. The first thing is the acronym, just 3 acronyms. MEF, or you can say FEM, whichever one. Okay, what does M stands for? Meaning of inclusion. What does E stands for? Examples of inclusion. And then F stands for what? Functions of inclusion. What is the function? What's the relevance? Okay, all right, let's start with the meaning of inclusion. Now, as you look at the meaning of inclusion, you can see there inclusion sneaks. So it's not only human being that sneaks or animals, inclusion also sneaks. That's the way we want to look at definition of inclusion. Okay, now inclusion sneaks. Now inclusion is a suspended non-living inactive metabolic chemical substances in cell matrix. What's inclusion?
Is the inactive metabolic cell
Good. I didn't expect you can say that again. What's inclusion?
non-living inactive chemical substance.
That's right. Non-living inactive chemical substances. You can see non-living inactive chemical substances. Okay. Now, we are done with the meaning. The next thing is example. We use the acronym GML versus BCH. GML versus BCH. Okay. Now, what's G? It stands for glycogen. Okay. Found in liver. We said glucose is stored as what? Glycogen in liver. And does this relate to what's the meaning of inclusion? Yes. Because we said inclusion are suspended, non-living, inactive metabolic chemical substance in where? Cell matrix. Good. So M for what? Melanin. Okay, melanin is found in the skin of black people. Is it not true? Good. L is lipofuscin. This one is not so common, but we call it the wear and tear insoluble pigment from leftover lysosomal digestion found in neurons and cardiac myocytes. That's cardiac cells. Okay, they're found. You can see the picture here. All right. Wear and tear insoluble pigment. All right. Found in the leftover lysosomal digestions. Okay. So once again, what's inclusion? What's the acronym for inclusion? SNCS. What's the acronym for the examples? GML versus BCH. Okay. And you know what SNCS is? If you don't know, take your time again. Say it over and over. Say it with your eyes closed. Say it with your eyes open. Try and write it out from your brain. Say it one more time. And then you do the same with the example. Like that. Don't forget, when you learn one thing, you repeat it. You recite it. You rewrite it out. Then you add something new. You repeat it. You recite it. Then you write it out. Okay, like that, till you know everything. Are we together? Now, the next one is versus. Versus stands for vitiligo. You can see it on the skin. Vitiligo. You can see vitiligo on the skin. All right. Vitiligo is when you
made reference to it.
Yes, vitiligo, they are of 2 types. They can be a clinical case of it. They can be a physiological case of it. The pathological case of it is a clinical case where you see some people, part of their body is as if it's white. Or somebody that is black, it's as if some part have some tribal mark or birthmark. Good. That is what we call vitiligo. Now, the normal one is when melanin concentrates in a part. Then the pathological one is when some part of the body, okay, is having patches on them. Okay, now that's that. We are not really looking at the explanation. We are just after the what? We are just after the examples, just mentioning the examples. Okay, so GML VCH. Okay, G for glycogen, M for melanin, L for lipofuscin, V for vitiligo, and then S for Sertoli cell crystalloid. Look at it. Sertoli cell crystalloid, SCC. Okay, this one is found, okay, in testes. They form the basement membrane of the testes in males. Okay, there's another one there, Sima brown inclusion. These are found in macrophage. Okay, macrophage are formed from monocytes. Okay, and macrophage are part of the fighter cells. Okay, let's move on. So B is for
B is for bilirubin.
bilirubin. Bilirubin are found where? In liver.
Yeah.
So far, we have studied examples of inclusion. We want to quickly look at the function. Okay?
Yes, sir.
B is for bilirubin, C is for carotene, and then H is for what?
Hemoglobin.
Hemoglobin. C is for carotene, H is for hemoglobin. Carotene are found in white skin, people with white skin. Okay, now the function of bilirubin, you use once again another wonderful mnemonic, NDC. You can say NDDC. Using now NDC. So N for nutrient reserving, B for cellular shielding, and D for debris warehousing. So NDC, very simple. NDC, very simple. Nutrient reserving, debris warehousing, and cellular shielding.
NDC.
Yes, NDC. Okay, NDC. So as nutrient reserving, number one, it acts as temporary storage for, remember what we said, for glycogen.
So that NDC is for
N-
function.
yes, function. NDC is for function, nutrient reserving, cellular shielding, debris warehousing. Nutrient reserving, cellular shielding, debris warehousing. Okay, so in summary, we have learned three things, three acronyms for three in-depth internalization.
Nutrient reserving.
Nutrient reserving, cellular shielding, and debris warehousing. See it now. Debris warehousing. That's the inclusion serves as trash can, debris. Trash can for debris. Okay. Metabolic debris. Metabolic waste. Okay. Can we move on from there?
So
All right.
center L versus BCH.
Good. So now we've learned three things about inclusion. Three important things. The first is the meaning with its
That's
acronym. The second is the example with its acronym.
GML versus
And the last is the function with its acronym.
NDC.
Okay, meaning
NDC. Nutrient
reserving.
reservation.
Good. D for what? debris
debris,
warehousing, then C,
cellular.
shielding. Okay, let's do that again. That's giving us headache, so we do that again. All right.
That
Functions: N is for what?
nutrient reservation,
Nutrient reserving. Okay. D is for what?
debris
Warehousing.
warehousing,
And then C is for what?
cellular
Shielding. Are you seeing it now? Now look at it this way. Look at it this way. Inclusion shields the cell from being harmed. Number two, inclusion stores metabolic waste. That's debris warehousing. The first one, cellular shield, it shields, is like a protector. All right. Remember when we were talking about rib cage or thoracic cage in thorax, we say the thorax or the thoracic cage is what? Is a protector. It offers protection to what? The visceral organs. Is it not true? So that's the same thing with inclusion. Inclusion has three functions: NDC, nutrient reservation. It reserves nutrients from flying out of the cell. You seeing it now? Okay. Number two, it trash debris, that is debris warehousing. All right. Make sure debris from metabolic waste, okay, is not scattered and littered in the body system. Are you seeing it now? Then the third one, inclusion shields the cell. If someone walks under the sun does not, turn to something else because of melanin. Are you seeing it now? All right. So that's where we'll end it here. But before we end, we run through it again. What's the acronym for the function of inclusion? NDC. What's the acronym for the examples of inclusion?
GML versus
GML versus BCH. That's correct. What's the acronym for the meaning of inclusion? NICS. Good. Now we look at the meaning of each of those acronyms. For the functions of inclusion, what is the meaning of NDC?
N is nutrient warehousing.
Nutrient reservation. Good. How about D is for what?
D is for debris warehousing.
Debris warehousing. How about C is for what?
Cellular.
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 How did I explain that cellular warehouse? Cellular dash.
Cellular. Shielding.
It shields, it protects. That's another way. When you try to explain it, it saves you from cramming. Are you seeing it now?
Yes.
Nutrient.
Nutrient reservation.
It stops the nutrient. Imagine you eat now and the food fly out from your system. Are you seeing it now? So inclusion does what? It stores it beautifully, it reserves, it stores it. And it stores it, for example, as what?
Yes.
If it's carbohydrate, it stores it as what?
Glycogen.
Are you seeing now it relating with the example? Good. Because when you are studying, you should be able to store it by connecting. You connect the function to the example. Are you seeing it now? We move on. Cellular shielding. Cellular shielding. Which example of inclusion can act as cellular shielding? Think. We have glycogen, we have vitiligo, we have melanin, we have lipofuscin. Melanin because it's on the skin, black pigment. Okay. Yes. And then vitiligo too. All right. Can also shield. Are you seeing it? Nutrient reserving. C for what?
C for cellular.
What does it do to the shell? Yes, cellular shielding. It shields the cell. And then D for what? D for debris warehousing. Let's look at the examples of inclusions again and ask ourselves, which of these examples of inclusion can act as debris warehousing? All right. Could it be bilirubin? Yes, because bilirubin, okay, is one of the structures that make up a bile. All right, make up bile, and bile is a excretory product. Okay, it's an excretory product. It's stored in gallbladder. That's what responsible for coloring the fecal matter and coloring the urine to be golden yellow and coloring the fecal matter to have those colors. If not, fecal matter will be pure white. Do you follow? Will be pure white.
So I don't know the fecal matter. Is it the skin?
Fecal matter. That's feces, excreta.
Oh,
Yes. We call it fecal matter anatomically. All right.
yes.
Okay. So that's a wonderful one. So we've done that. So we are going to make another video. Okay. As if we never internalize this. Okay. So that's what we are saying. It's a session or a channel where we internalize concepts in piecemeal, little by little. Now, once we have internalized it, other sessions will be running through it as presentation without what? Without much without much without much scratching of it, because we have done that in internalization sessions, and we keep doing till it sticks, so that even when we want to forget it, we find it difficult to forget it. Even when we are finished writing exam, we find it difficult to forget it. All right. So see you again in the next session.